Hey everybody, this is Dr. Cummings from Microbio. In the chemistry review quiz that you took last week, the most common questions that were missed all had kind of a central theme around them. They all had to do with understanding hydrophobic, hydrophilic properties of molecules and how their hydrophilic or hydrophobic nature would cause those molecules to behave. So I want to go through 3 of those questions and see if I can clarify what's going on here a little bit. So the first question that was relatively commonly missed asked about a lipopolysaccharide molecule, and it said that the lipid portion of LPS would be found where? So in order to think about that, we've got to first remember what a lipid is, right? A lipid is, by definition, a hydrophobic molecule. Hydrophobic molecules don't want to be around water. There's nothing to them that attracts them to water, no charges, no polarity. And so they're actually going to try to hide and bury themselves someplace that's also hydrophobic. So we've got these molecules, these lipopolysaccharide molecules, that are going to bury themselves on the interior of this outer membrane in the gram-negative bacteria, right? Here is your lipid A. And connected to that lipid A is going to be a polysaccharide that likes water. So this is hydrophilic, and that's great because out here you've got a lot of water. And your lipid A is hydrophobic because this entire interior of a phospholipid bilayer membrane is entirely hydrophobic. That's where we find a lot of hydrophobic molecules. So in order to answer this question about where would you find the lipid portion of LPS, you've got to remember that lipids by definition are hydrophobic, and since it's associated with a membrane, it's going to hide itself in this hydrophobic interior of the membrane, allowing the polysaccharide chain to splay out into the hydrophilic extracellular environment where there's lots of water. Let me show you an image from the textbook that shows this a little bit better. So this outer membrane here, from here to here is your outer membrane. Sometimes we call that the LPS layer. And you've got a hydrophobic interior to it. You've got at the surface hydrophilic molecules on either side that are going to want to interact with water, and that's good because the region in the periplasm is aqueous, meaning it's water-based. The region outside the cell is aqueous, meaning it's water-based, right? And so the outer regions of the membrane are going to want to interact with water because they have charges and polarities, but the inner region is going to be entirely hydrophobic, and the only thing allowed in there will be hydrophobic molecules. And so an LPS molecule, if we go over here to the left, here's an LPS molecule, and they're saying that this is your lipid A. And your lipid A has to be locked into the interior of the membrane to avoid contact with water. And that allows the polysaccharide then to wave out in the breeze and interact with all the H2O molecules, because polysaccharides are going to be highly hydrophilic because they're made up of a bunch of sugars. Okay, hopefully that helps to explain a little bit of that question. Now, another one that was really similar said, we gave you the picture of tyrosine. We said, okay, the R group shown in green above in tyrosine, what impact does the hydroxyl group have on how this amino acid will fold into a polypeptide chain? Remember, a polypeptide is one amino acid linked to another amino acid, linked to another amino acid, linked to another amino acid, on down the line for hundreds of amino acids. They each have a carbon and a central carbon, carboxyl, an amino group. They've got a hydrogen coming off. The peptide bond that you see here connects the amino group of one to the carboxyl group of the next. And coming off of each of them is what we call their variable R group. And it's the different R groups that give each amino acid their characteristic traits. And based on these R groups and the pattern of these R groups, they're going to want to fold the final molecule into some sort of a structure that allows R groups that are attracted to each other to come together, R groups that hate each other to be pushed apart, R groups that are hydrophobic to avoid water, and R groups that are hydrophilic to come into contact with water. So the R group for tyrosine is this box here, much of which is relatively hydrophobic, but the question says, what about this R group, this hydroxyl? And if you remember, a hydroxyl is an OH, and the O being highly electronegative is going to have a delta negative, a partial negative charge. And the hydrogen will leave essentially a partially exposed proton with a partial positive charge in equal magnitude to the partial negative charge around the oxygen. And water just loves that bond, because water, if you remember, is HOH, more OHs, right? And so you've got partial positives around your H's, you've got partial negatives around your oxygen, and they're going to want to interact with each other and anything else that's charged or polar. And so this residue up here, this OH functional group in tyrosine, if at all possible, is going to want to help this total molecule to fold in such a way that the OH is ultimately exposed to water. In fact, residues like tyrosine are often found on the outermost surface of a protein if that protein is found in an aqueous environment, because all this would be water all the way around. And so all of these amino acids would need to be hydrophilic in some way, because if they're going to be able to interact with water. So the final question then, also related, says, what kind of amino acid residue would you find on the exterior surface of a membrane embedded protein? So now we've got a protein, like this glycoprotein here, and we're asking about this outer surface here that's embedded in the membrane. So now we've got an outer surface amino acid that is in contact with this hydrophobic interior of the membrane, and therefore these amino acids are going to have to be hydrophobic amino acids, because otherwise they would not be welcome in that interior and the whole protein would have to get popped out again if they were hydrophilic, if they were charged. Same is true on this surface out here that's exposed to water. That's going to have to be hydrophilic. But in the case of this question, it's asking this, the exterior surface of a membrane-embedded protein. So we're talking about this surface here, embedded in the membrane. It's going to have to be hydrophobic amino acids. So think through those principles, because now we're not just asking you to memorize what it means to be hydrophilic or hydrophobic, but to take those concepts and try to predict the way that certain important biological molecules are going to behave based on whether or not they want to interact with water. Really, really important principles for these macromolecules in microbiology. And as always, let me know if you have questions.